Okay, we have a few more responses. So Zineb has no experience in bioinformatics, genomics, but is interested in studying clinical genomics of infectious disease. Interesting. Vash Wasim, okay, sorry. Recently defended PhD Viva Voice in animal biotechnology. Suleiman is a Pakistan graduate with medical laboratory science, further going for public health. Okay, so we have a diverse range of participants from molecular biology, genetic engineering, chemistry, animal biotechnology, veterinary sciences. It's good to see so many diverse participants. Okay, Ishrat is interested in data science, plant biology. Thank you. Okay. All right then. So without further delay, I'll slowly start sharing my screen and then we can start with the presentation for today. So I'll slowly start with a brief introduction to the team, who we are, what we do, and also a little bit about the platform where we have a comprehensive collection of resources that might be of interest. I'm also receiving some request for a recording. So all of this will be recorded and we'll also share with you the link via email. So it might take maybe by tomorrow, you will receive all of this in your inbox. So just keep an eye out for it. And with that, I'll now start with the recording once and share my screen. Okay. Okay, so we'll try to keep this webinar interactive as much as possible. And it was once again so much, thank you so much for everyone for introducing yourself in the chat box. And it's always good to see participants from across the globe getting interested in bioinformatics. And we get a lot of queries and interest from participants who are interested to either transition to the field of bioinformatics or they're already in the field of bioinformatics and would like to specialize in a specific omics field further. And there are also a lot of students who are interested in working on research projects in bioinformatics. So we'll be covering all of these on how you can get started in some of these topics and also what are some of the opportunities that we provide at OmicsLogic. Okay. And so I can see some familiar participants who have been previously maybe part of our workshops or maybe part of our previous webinar, but also we have a majority of participants who are joining with us for the first time. So for them, I would like to take a brief moment to introduce you to the team behind the webinar. So we are a global bioinformatics and data science company with roots in India and the US. And over the years, we have had the privilege to collaborate with leading universities, research institutions, and industry partners worldwide. And our mission is to bridge the gap between data driven research and skill development. To achieve this, we work with regional and international experts who help us tailor our programs to the specific needs of students, educators, and researchers across the globe. And now, with the growing demand for skilled bioinformaticians and data scientists, we have developed a wide range of training and research offerings, including online courses, specialized workshops, internships and fellowship, research services, as well as a degree program. Now, here's a quick overview of the student learning journey that begins with hands-on courses and programs that is suitable for all participants from diverse range of backgrounds. So even if you're a beginner or even if you're an expert, there is always a step for you that you can get started. Now, like I said, we also get a lot of interest from participants who are interested in doing research. So we also have a research program that provides a platform for the participants to work alongside a mentor and complete a research project. Now, we also offer courses and example projects on a diverse range of topics from genomics, metagenomics, transcriptomics, epigenomics, GitHub, Linux, as well as application based projects. We'll be covering all of this in just a while, but I just wanted to introduce everyone to all of these just to help you get an overview of what's coming next. And all of these application based projects are based on several topics like infectious diseases, agribioinformatics, oncology, neuroscience, and so much more. And all of these courses, they are enriched with engaging content, including visuals, infographics, and video lectures, that simplify all of these complex concepts for better understanding. And we also focus heavily on experiential learning through short tutorials, practical demonstrations, coding exercises, so that the participants, they are able to build technical expertise and the confidence. Now, while all of these courses, they are fully automated and lesson based, we also understand the value of mentor guided instruction. So that is why we also offer workshops and training programs that combine instructor led sessions with hands on walkthroughs to reinforce key concepts. And we will be exploring once again all of this in through our live online platform as well. And this includes a combination of expert video lectures, interactive tutorials, and step-by-step lessons. And all of these are curated by mentors across the world to ensure that the learners are able to get best of both theoretical knowledge as well as the practical application. And here's a quick glimpse into some of the feedback that we have received across the workshops and programs that we have conducted. A lot of the participants have gone on to went and post on social media of the certifications that they have got, the feedback, and so much more. And I'll quickly stop sharing and take all the participants to the OmicsLogic platform so that we can slowly get started with the introducing of the platform. I'll reshare my screen once again, just a moment. So some of you might already have an account on the platform if you have previously been part of any of our workshops or some of the webinars. But for those of you who are joining with us for the first time, let me share the link in the chat box once and also show you the steps on how to log in. Okay, Rajni has already shared, and let me share my screen. Okay. So if you click on the link on the that is provided in the chat box, you'll be redirected to a similar interface, which is an online learning platform where all of the study resources that I was talking about in my slides just a while ago are available. And we'll be expanding more about that by the mentor who has been actively working with the students on improving several of the resources to make it accessible and easy for everyone to get started. Now, it is a very simple step to create an account on the OmicsLogic platform. It is free. All you have to do is just head over to the platform and click on login. And once you click on login, you can click on create an account and you can enter your name, email ID, password, and you can create an account. Or if you already have an existing social media handle, such as Google, Facebook, Apple account, GitHub account, or even Twitter, you can sign up using any of these social media handles as well. Okay, and for those of you who already have an account, simply enter the existing email ID, password, and you can directly log in. Okay, so this was just a brief introduction that I wanted to give you. And with that, now I'll pass over the stage to the mentor, Ritika Patyal, who will be guiding you through all of these various resources that we have put together for the participants to explore. So over to you, Ritika.
Okay, thank you, Shrikari. Good evening, everyone. Okay, so as demonstrated by Shrigori that how you can create your account once that is done. So I'll take you to the portal and I'll tell you what are the different products that are available that will help you to start your journey. And if you're someone who's already familiar with the concepts of bioinformatics, so how you can take it to the advanced level, or you can move towards your own research project also. So once you have created your account, here you can see we have several courses available. So as discussed that we have a course category for bioinformatics. If you are someone who is starting its journey in bioinformatics, so this is the course that is specifically designed to start your journey. Then if you're interested in genomics, then transcriptomics, machine learning, Python courses there, courses which are specifically designed for omics analysis, metagenomics, cheminformatics, single cell RNA-seq, epigenomics, Linux, and GitHubs. So these are several courses that are available on the platform that will give you the brief introduction. So it has been divided into the 3 states: beginner, intermediate, and the advanced level. So I will open one for you and I'll tell you. So from the repository here, you can see if I click on any of it, this genomics. So I want to explore this. So according to the category that you're going to choose, there are several projects also. So these projects are available along with the codes so that you get the detailed, or we can say you get the a research experience of different projects. What are the research questions that can be answered? So you can see this is an entire repository for genomics course as well as the projects that are associated with it. So I've opened my genomics course like this. So this is a detailed end-to-end genomics course, which has several lessons over here. I'll show you each of them and what content is present in each of them and how it will help you to understand the concept of genomics from beginner to the advanced level. So if I open my first lesson, in which we are talking about DNA structure and variations. So this is here, you can see this is all textual content is here, where we are talking about the basic concepts in order to understand what the genomics is all about. Here you can see we have integrated short video tutorials are also present. The updated content is present. What are the learning outcomes? Here you can see we have a summary of the program. And then at the end, you will find the quizzes, which will help you to revise that what you have learned, you were able to grasp that, right? So this is all textual content that is present in the lesson. If I move to the second lesson, which is about the genomics explained. So in this, we are taking it further. Here you can see the learning objectives is all about the different sequencing technologies are there. So this particular lesson will give you the demonstration of what genomics is, how sequencing, what type of different sequencing platforms are available, what are their applications, what is the right approach or what is the right way to go about the sequencing platform if you are choosing a correct research question. So again, the textual content with the short video tutorials, they are elaborated in the lessons over here. And here you can see we have summary and then different types of sequencing platform, ghost technologies. So everything that you will find here is going to add something to your knowledge. Apart from this, we have in our lessons, we have integrated codings also. So in coding, we are talking about, here you can see this lesson particularly talks about that how you can make use of R programming for genomics analysis. So we are starting with the way that you, what prerequisites are required, what types of softwares are required, how to go about the installation. So everything is in a way that if you're a beginner, so it will help you to start your journey in an easy manner. So here you can see in the lesson here, the coding scripts are available. This is our script, what packages are required, and then what our commands are required over here. So everything is elaborated in this particular section. And if I move further, so here you can see how the input is, what are the different file formats. So in genomics, we talk about the FASTA files, we talk about the FASTQ files. So different types of file formats, how to give the input, what kind of translation analysis can be performed, multiple sequence, if you're working with multiple sequences. So this is the lesson that will help you to understand the utilization of R in your genomics journey. Apart from this, in our lessons, we have infographic visuals also that will help you to understand the concepts here. So here you can see we are talking about the wet lab aspects. So we are starting from the very beginning. We are giving you the overview of the sequencing, how it starts, how you extract the DNA extraction techniques we are talking about here, then quantification techniques, spectrometry, all the gel electrophoresis. So all the technology, all the steps that are required before bioinformatic analysis, they are also here so that you understand that how the data was prepared, and what type of analysis has been done. And then here you can see, along with the video tutorials, there are infographics here, which this is all about the Sanger sequencing and what are the advantages, limitations, what are the different sequencing platforms are available, what to choose, what is your research question. According to that, there is a detailed elaboration of the sequencing platforms in the lessons itself. After this, if I open the lesson 5th over here, okay, so this lessons here where we're talking about genomic sequencing data output and download strategies. So here, you will get familiar with the different file formats that are used in genomics analysis. So here you can see we are starting with the FASTA file, what the FASTA file is, what it looks like, what are the different information which is present in the FASTA file, what is a SAM file, how does it look? It's a text-based file. So the entire information of all the file formats, SAM file, BAM file, GFF format, where they are used, GFF format, VCF file. So you will get introduced to the different file formats so that you know that at what particular steps these files are generated and how you can how can you use them for your analysis. Then there are some tools that are used. Map files is there. Then a map file from the TCGA platform. This is a very well-known platform if you're someone who is working with cancer data set. Then you get familiar with the repositories from where these files can be accessed, FTP formats, FTP usage, the API interfaces. You will get familiarized with them. And then you can get understanding about the cloud platform. What are the different cloud platforms that are available for the genomics analysis, and what types of applications are present over here. So this is something that you can learn from the particular lesson. Then, apart from this, there are several interactive visuals also present in between the lessons. Okay, so like this, here you can see this interactive visualization will help you to understand the entire genomics analysis workflow, that what are the different types of analysis that can be performed. For example, this is very helpful for someone who is looking at genomics analysis or genomics pipeline for the very first time. So you start with, after you are done with your wet lab aspect, you are going to learn it from the previous lesson. You start with the sample collection, library preparation, sequencing platform, and once you have the raw data, what can be done with that? So here you can see you have a pre-processing and the quality control, what steps can be performed, what are different tools that you can make use of. Then you have a decision to make that you want to go with the reference-based or de novo-based. Then according to your choice, if you are going with the reference-based alignment, then what are the different tools and what are the different steps that you have to follow. Then if you're going with the de novo genome assembly, de novo is usually when you do not have a reference genome or when there is a novel organism in your research, then you go for the de novo genome assembly. So these here you can see the different steps, different tools, and different file formats that are used are explained over here. Then genomics analysis type, you want to perform germline variant calling, somatic variant calling, population variant calling, gene expression study, epigenomics, structural variant. So all types of analysis that can be performed are elaborated here. And then for the variant annotation and the functional prediction, so these are different tools and different resources that you can access. And then you can find a specific application also in the clinical oncology, microbial genomics, clinical genetics, agriculture genomics, evolutionary bioforensic genomics. So this is how we have designed our courses, where with the text, with the videos, with coding, infographic visuals, and also the interactive dashboards that we have, along with the interactive activities which are present in some of our lessons over here, which will give you an overview or which will give you experience of working with the genomics data. So here you can see this lesson, it talks about the de novo assembly. So here you can see we are talking about the quality control, the fast QC, what are the outputs, how to use the tool, what outputs you are going to get, what are the different tools, and what is the algorithm, what each command is talking about. So everything here, it is given in a very detailed manner that, for example, you are working in Linux for the first time. So the step is like that: you have to create a conda environment, you have to set up the environment, and you have to work with installation of the tool. So everything is given in a easy and a follow through manner, like what types of how much of RAM is required, how much of CPU cores are required, this space is required, which operating system is required. So all of these things, they are provided in the lessons over here. So this was just an overview here, and let me tell you that this is a comprehensive course. It is very easy to go through and anyone can do this. So it's not like that it's very difficult to follow. It's just you have to be, you should know about what technology you're going to learn, and this will help you to take you to the your better understanding. Now, I was showing you here, for example, you have completed your genomics train this entire course over here. Now you want to utilize this. So in order to utilize this, you can go to any of the project. For example, this is one of the project of COVID-19, origin and pathogenesis of SARS-CoV. So here you can see the project is in such a way that first the case study is explained, that what the research question is all about. So this is all about the coronavirus, what coronavirus is, what is the incubation period. So first, it goes about the introduction of the case study. Then it tells us about the genomics resources that are going to be used in the particular project, so that before moving to the actual hands-on part, you are ready with the setup, you are ready with the data also. So here you can see it is talking about, after explaining the research question, what type of data you can make use of. So this is NCBI virus. From here, you have to download the particular data set that you can practice. Then what are the different tools that you are going to require, the Fig tree, Eugene, Chimera X. So you are going to get started with your setup over here. Once you're done with this, once you understand the question, setup is ready, then you move to the third part where you perform the steps. So first step is to retrieve the sequence. Then the explanation is also there, what kind of output is going to be, how you are going to follow the steps. So it is given in easy steps over here, that you have to follow the step. You have to build a phylogenetic tree. Then you have to click on the particular format that you're going to get. And then the outputs are also given, that this kind of output you are going to get and what is the conclusion for that. So biological interpretation is also provided, so that not just the analysis, the biological significance is also what you understand. Then the next level of the project is about the multiple sequence alignment at the nucleotide level. So here you can see how you are going to, which tool you are going to make use of. This is Eugene. You are going to download it from the official site. What steps you are going to follow over here. You are going to load the sequence, and then you're going to perform the analysis. And this is what your output is going to be and how you're going to interpret that. So this is how our projects are designed, so that after learning about the particular technology, you learn the application of the particular technology in various aspects. So here you can see we are talking about the comparative study over here. So here also we are talking about the methodology, then the phylogenetic tree construction, and then the interpretation is provided. So this is one of the project which is on COVID over here. Let me open one more project for you. So here you can see okay. So this is a brain metastasis genomic from mutation to medicine. This is also designed in a similar way, where we are talking about the project overview, then we are talking about the setting up of the genomic analysis toolkit, and then in the project itself, once you are done, once you understand the research question, you have the data ready, then you talk about the analysis. So here you can see again, this is the R code that is provided for creating. This is a MAF visualization. This is a mutation landscape. So you load the data after understanding the research question, as these are different types of outputs that can be generated. And these are along with the biological interpretation, that what the each step means and how you can interpret it and how you can make use of them in your own study. So you can see, if you are someone who is already working in genomics, so you should know that this plot is giving us the mutation insights, the hotspot regions it is defining, and what type of mutations are seen in a specific position. So this plot is highlighting that thing. So these are for specific genes over here. And then there are some advanced visualization also that can take up your research to next level, because adding the visualization is a better aspect to representing your results over here. So this was all about courses and the different projects. So I've only showed you the genomics, but if you see, this is the way how we have kept our, for transcriptomics also. For example, in transcriptomics again, similarly, you are going to have an entire course where you're going to learn about the basic, the beginner level is there, then you talk about the technology, then after introduction to the technology, you move towards the visualization. So here you can see you have statistical tests are there, a workflow of the differential gene expression analysis, functional enrichment analysis, DEG analysis in R. So this is how each of the technology has been designed, along with the courses and the several projects are available here. So you can see this is a TCG liver cancer precision oncology project. Then a Python course is available. Then BioML machine learning. So these are different courses that you can access along with the projects for the research experience. Okay. So now after this, if you're someone who want to learn about these technologies, so we have training programs available. So these training programs, they give you access to the material. For example, if I was showing you genomics here, so I can show you genomics training program, which will give you the full hands-on experience of the technology from beginner to the intermediate level. So this is how it should be once you have created your account. But if you do not have access to this, this is how it should look for you. So if you are someone who is who wants to see that what material will be available, so you can see this is a beginner level. Then in the beginner and the intermediate and the advanced level is there. So in each level, we are going to have several sections over here, like introduction, then Python tool. So this is how you are going to get the visualization, that what each lesson or the each session is going to be about. So you can see we are talking about the laying of the foundation over here. Then we are going to talk about the tools and everything, and then the lesson fourth. So this is how it moved towards the next level. So in the particular, I have opened the NGS Genomics Data Analysis Training Program over here. So in the training program, you are going to get the theoretical concepts along with the hands-on experience also. So you will learn about the technology, you will learn about the concepts, and then you will get the collabs and resources that you can again make use of them for your learning experience. So here, for every session, you are going to get a video. A video tutorial will be present, which you can easily go through. You just have to play it, and according to that, what are the associated lessons. So here you can see Introduction to Bioinformatics course. So you can easily access this course from here. So with every session, if there is a lesson to be reviewed, so this will be added for you. If there is additional reading material, any research paper has to be added for the better understanding of the concept, then it will be added as a resource material over here. So once you complete your one level, then you move to the next session over here. Like here you can see we're talking about genomic variation, then role of genomics data analysis, and then we are talking about the technologies over here, then different file formats, then phylogenetic analysis. And with this, you can see multiple sequence alignment over here, the DNA replication and the reverse complement in R. And you are going to get access to Google Colabs also. So Google Colab is a notebook where you can perform your coding here. So this is a Google Colab in R. If you want to, if it is in Python, so you can easily change it to Python also. So this is how you will get the access to Colabs over here, which you can easily practice. And you are going to get the outputs also, and the explanation of outputs, you can get it from the video tutorial as well as from the lessons itself. So this is how then VCF is there. So this is how the each level is explained. After your intermediate, you move to the advanced level. So this is how every material is present from each level. So if you are familiar with the beginner concepts, you can move with the intermediate, and then if you are familiar with the intermediate, then you can move towards the advanced level where you can learn about the reference-based assembly, Linux, if you're someone who wants to learn genomics in Linux. So this is how you can take further up your journey in the bioinformatics. So once you have taken up the courses and then you have completed your training program, and if you want to have a further experience of research, or you have a research question in mind, or
good afternoon, everyone.
Yeah, hello.
For 3 minutes to
Sorry.
Yes, Ritika, you can go ahead.
Okay. So while through the learning process, if you get interested in a specific paper, or you want to replicate a paper, or if you have a research question of your own that you want to take up, so there is a separate research program that is available where you can work with a mentor, and you will be guided through every step. The materials will be provided for you. If you want to take up your own research, then the help will be provided. If you want to work on a project that the mentor will be providing for you, giving the data set or selecting the topic for you, that can also be done in the research program. So Shri Gauri will take up further to explain what the research program is all about. So over to you, Shri Gauri.
Yes, thank you so much, Ritika. Let me share my screen.
Yeah, Shrigari, if we can take a few questions now, because there were a few questions, so maybe take this as an opportunity to do that.
Okay, so I'll start from the beginning. Certificate for joining webinar. So, Sharif, whenever we are providing certificates, it's usually after we assess the students, like through quizzes or some kind of hands-on practicals are involved, and they submit whatever they have learned through the workshops or training programs. So it is through those that we usually provide the certificate, that is courses or workshops or training programs or research project, so that we can verify that yes, the participant has gained skills after completing their training, and that is when we issue the certificate. And there is another question, sign up is free, but what about the courses and resources? So I'll quickly share my screen. So there are courses and resources that you can go through. So here on the screen, if you head over to the courses page, and if you see the first one, bioinformatics, and if you click on explore, you'll see the very first course, course 1, Introduction to Bioinformatics. You can click on that, and it will redirect you to this very beginner friendly, easy coursework, which is free, that all participants can go through and get a certificate once they complete the required quizzes and assignments at the end of each of the lessons. So it's very beginner friendly. That introduces you to what is bioinformatics, how they are applied in the field of health sciences, and also will introduce you briefly to genomics, transcriptomics, and metagenomics, that is various omics field. Just make sure that you click on the load more button. Sometimes participants tend to skip when they are going through the coursework. They tend to skip some of the lessons that are there at the end of the coursework. So just make sure that you click on the load more, and once you click on load more, you'll be able to see the rest of the lessons within the coursework as well. So it is a very comprehensive coursework, very beginner friendly and introductory as well. You can go through it at your own pace, get familiar with the topic. And once you go through this coursework and you get a certificate, and if you're interested in further specializing in a specific omics field, then you can opt for those training opportunities that Ritika was talking about in her presentation just a while ago. I hope that answers some of the questions that has been posted in the chat box. And is there any scholarship? Jean, we do provide some scholarship opportunities for participants for the training program and the research program combined. So I'll be explaining that in just a while towards the end of my presentation as well. So first, we wanted to cover the training opportunities, and now we have some research project opportunities that those participants who are interested to enroll, we can discuss about that as well. Rajni has shared the link in the chat. Samuel, is the certificate free of charge as well? For course 1 Introduction to Bioinformatics, yes. I've shared the link also once. You can go through it. It's beginner friendly, and at the end, you will be able to get a certificate on your profile. You can head over to this and click on dashboard and head over to certificates, and you will get it. And please feel free to tag us on social media once you get a certificate. We do have a community of around 35,000 plus researchers, faculty, students in our bioinformatics community, and they all are happy to see the achievements that you share in the community as well, and we'll be happy to repost. Okay, I guess that is all that I can see in the chat. I've answered most of it. And now I'll just briefly take all the participants through the research program quickly. So in the beginning, I have briefly explained about the program, but I can see that some participants have joined a little bit late. But for those of you who are joining late, I'll just quickly give you an overview. So we also provide a research program opportunity that provides a platform for the students to work alongside the mentor on a bioinformatics research project. And with mentor support, this program will go beyond traditional learning, where you will be delving into real-world bioinformatics research using cutting-edge omics data. And one of the most frequently asked questions is, how does this research program work? So we have a roadmap here that explains the entire journey that we take the participants through, right from beginner. That is another recurring question that we get, like, I am from a biotechnology or from a biology background, so can I work on the research program? So yes, it is completely beginner friendly. We will start with the training, and it will end with a project submission. I'll take you one by one through all of these phases as well. So here on the screen, you can see the full roadmap once. So it begins with the students undergoing comprehensive training in their chosen field of omics. So this could be genomics or transcriptomics or metagenomics, metabolomics or cheminformatics, all of these omics field that Ritika was talking about in her slide. And the objective here is to help the participants build a strong foundation in key skills such as data processing, data analysis, and data visualization. And in the next stage, students will start working on identifying and impactful research topics. So this will be conducted through regular mentor meetings. There will be meetings that will be scheduled for you once a week, where you can interact with the mentor and work on the project topic, and also followed by through that in-depth literature review. And now in the third step, students will start working on data collection, analysis, and interpretation. And this is also the stage where students will start looking into data set to identify trends, patterns, and correlations that will be very critical for their project. And finally, the program culminates in students working on completing the report, detailing all the analysis and interpretation they have carried out, preparing presentation and submitting the work so that they can receive expert feedback to refine their work. And now I'd like to also quickly take you through how students from our previous batches have leveraged these learning opportunities that we have presented before you, as well as the research opportunities to achieve their academic and professional goals. So here on the screen, you can see a cohort of students from Systems Medicine graduate program who completed their initial training in Python and machine learning and went on to later work on research projects on various biomedical research challenges such as infectious diseases, agri-bioinformatics, metabolic disorders, and so on. All of their projects are also available on our platform. So I can show you in just a while at the end of my screen as well. So you can also take a look into some of these projects that have been submitted. Students have also went on to present their research projects in poster presentation competitions to win exciting cash prizes. So here on the screen, you can also see a student from high school background who completed their transcriptomics training and worked on an Alzheimer's disease project. He later went on to present at the Regeneron International Science and Engineering Fair, for which he won the Potemkin Prize, and he is currently accepted into the Harvard University for chemical biology undergraduate degree as well. And students also join the program to get research publication done that gives them this extra competitive edge over other students while applying for higher studies or in the job market. And one of the unique highlights is that faculties and researchers have also collaborated to carry out computational research work, and it is also possible to pursue an entrepreneurship and business innovation track like a team of industrial researchers where they developed a microbiome test kit to improve the fertility of women in Nigeria. And now here's some of a quick glimpse into some of the preprints and publications that have been completed by our students in different fields, including stem cell and development biology, space medicine, and radiation protection. That is also an interesting area that students are keenly looking into. Cancer research is also one of the most popular amongst the students. Neuroimmunology and neurodegeneration, vaccine efficacy and immune response analysis, antimicrobial resistance and global health, COVID-19 and comorbidity risk, reproductive health and predictive modeling, structural biology and cheminformatics, metabolic disorders and diagnostic biomarker discovery. So one interesting aspect of this project is that it has been completed by a high school student, a preprint that he is now currently working on publishing it in journals as well. Now, as we are starting with the new year, and we have recently now opened the cohort for new students to start enrolling in some of the project opportunities that we have launched, where you can work alongside the mentor and complete it. And these are some of the ongoing project opportunities that I would like to present in the next couple of slides that participants can join in. We already have the project topic, and you can join in some of these projects and work alongside the mentor. And at the end of my presentation, I'll launch a poll where you can select the project topic that you are most interested to work on, okay? And just make sure that you select the correct poll, and then our team will connect with you further for further details about how to enroll in the program and also about the scholarship opportunities. I saw one participant was asking. So all of these our team will be getting in touch, okay? So I'll just start with one of the very first project, which is virtual screening for drug target, which is a 3-month project, and which is basically the objective here is to integrate omics data for the identification of disease-associated molecular targets and perform structure-based virtual screening to discover potential lead compounds. The second project is on transcriptomic profiling in systemic lupus erythematosus, and it can range between 2.5 to 3-month project. It is a transcriptomics project where you will work on identifying differentially expressed genes and dysregulated immune-related biological pathways. The third project is on multi-omic analysis in cancer. And here you will be integrating genomic, transcriptomic, and epigenomic data to identify key molecular signatures and pathways. This is also a 3-month project. The next is a machine learning project for identification and validation of predictive biomarkers and ranges between 2 to 3 months. And the objective here is to apply ML approaches to high dimensional omics data for identifying and validating predictive biomarkers that is associated with Alzheimer's disease. And the final project is on immunoinformatics. And here you will be working on designing and evaluating on a multi-epitope subunit vaccine targeting the dengue virus envelope protein using immunoinformatics and computational modeling approaches. Okay, so these are some of the opportunities that you can join in. Now I'll launch the poll in case you would like to work on a research project that is not mentioned here. Maybe you have your own lab generated data and or maybe you have a different research topic, then you can mention that in the poll. I'll keep the poll open for a couple of minutes. So for those who are interested to work on such projects, please select it. I'll also read out the poll questions so that in case you're not able to see it or read it properly. Okay. I have launched the poll now. Now on the screen, you might be able to see a pop-up that tells a webinar poll. So I'll start reading the fields one by one. So the very first section is to enter the email ID. So here, make sure that you add the correct email ID in the section that you prefer our team to contact you. Sometimes participants tend to add a wrong email ID with wrong spelling error. So please ensure when you are filling it out, just make sure that you are correctly adding it so that our team can reach out properly. The second section is to enter your research interest. So right now I presented around 5 project topics, and these 5 project topics have been listed in this second question. So the first one was virtual screening for drug target discovery. Second is for transcriptomic profiling in systemic lupus erythematosus. The third is multi-omic analysis in cancer. Fourth is in machine learning for identification and validation of predictive biomarkers. And fifth project was immunoinformatics. So please select a topic that you're most resonating with. You can select multiple one as well if you're interested. And finally, the third is optional, and it is for those participants who are interested in working on a topic that is not mentioned in the project topics. Suppose, let's say, something related to astrobiology or some other topic that you're interested to work on. Please mention that in the field, and our team can understand the questions better as well. Okay, so I'll keep the poll open for 2 more minutes, and then I'll close it. So I can see participants are slowly filling it. And meanwhile, I can also check the chat box for any questions that they may have. Okay, thank you. Rajni has also shared the link to the student project page where you can use some of the project that has been completed by the students. I can also share my screen and show you once. Okay, so here on the screen, if you head over to the project section, once you click on that link that has been shared in the chat box, you might be seeing a similar interface. And here you can see all of the student projects that have been submitted by our previous students. And it is very exciting to see project topics on different topics. And for example, there is one on multiple sclerosis, there is on cancer related, there is on agri-bioinformatics, machine learning related, all of these kind of interesting topics that you can get an idea. So I'll also open one of this just to show you the format of what it looks like. So this includes the submission from the student, a presentation recording of the project that they have completed so far. And these can be between 7 to 10-minute recording. And all of these projects are submitted in similar format that of a research paper. That includes abstract, introduction, methodology, results, discussion, conclusion, and finally references and acknowledgement. So as you can see, it's very similar to that of a research paper, and it is a great addition and a way for you to showcase your project portfolio, that all of the skills that you have gained throughout the training and the research program, it is a great way for you to showcase it to your peers and to your potential recruiters. You can add it to your resume. Anyone with the link to your project that you have completed, they can see it and understand the analysis that you have done throughout your project as well. So this is one thing that we wanted to cover. And in case there are any other follow-up questions that you would like to get in touch with our team and get a better understanding, our team will share their contact numbers in the chat box. They're also available through WhatsApp, so you can also drop us a message there. So I would like to request the sales team to just share their contact details once in the chat box. And if anyone is interested to explore any of these topics further, they can do so. And apart from that, if there are any participants would like to get understanding like of any specific training opportunity better, then they can also post it in the chat box and we can take up your queries one by one. Thank you, Rajni. She has shared the contact numbers in the chat box. Shall I close the poll? Any other questions? We can take that up. Okay, Kara has posted her contact number. Would you like a team to connect with you, Kara? If you can also expand about your query, that will be also helpful for the team to follow up easier. Okay, I'll close the poll in just a couple of seconds. Shall I close the poll? Anyone else is pending? You can see some of them are still typing. Okay. All right, let me close the poll. Until then, if participants have any questions related to the programs or coursework, please feel free to let us know. End poll. Okay. Anyone, any other questions that you would like to talk about? Let me also share once the link again to some of these resources that we were covering. One was about the coursework. I'll share this link in the chat box. And then we were talking also about the training programs. So these two are the links. And if, okay, there is a question. Shyam, thank you for doing this. I would like to learn genomics and transcriptomics. Thank you, Shyam. Can you also share your maybe email ID and our team can connect with you?
Hi. Can you hear
Yes.
me?
Yes, please go ahead.
Thank you. Actually, my question is actually what are the prices actually? Is that student affordable?
Yes, so our team will be connecting with you to further understand like what kind of training that you would like to go through. So you must have seen on the screen that we were showing different levels like beginner, intermediate, and advanced, and also the projects. They are of flexible duration,
Yeah,
like to
I got you. I got your point, and I saw you. I already visited your website, but I saw some. Some is free and some are not free. We need
right.
subscription. So is that student affordable?
Yes, yes. So we do make that student affordable, and we also provide discounts for participants who are from different backgrounds as well. So just get in touch with our team, and we'll guide you through the process as well.
Okay, so another question is, yeah, you already discussed, I think so. Maybe I'm, I missed it, I think so. You mentioned the project opportunities. So, is there available to join the project?
Yes, yes. So we have these project opportunities. They are completely online, but we do have limited slots. So just make sure that you select the project opportunity and you get enrolled at the earliest. And yes, we do have the program open right now, and we are accepting applications for it.
Okay. Okay. Thank you so much.
Yes, thank you. Any other questions? Let me just check. Sebastian, I entered the meeting a little late. What is the cost of participating? Yes, so Sebastian, it depends on the project that you're enrolling and also the training program the skill that you want to learn. So you can get in touch with our team. They will provide you the specific roadmap that is most suitable for you based on your interest, and then they'll let you know. So you can either drop a message to one of these contact numbers. I'm sharing it in the chat box once again, and we'll guide you through the process. Same way for Samuel as well. Sure, Marvit, we will get in touch with you for immunoinformatics project. Right, thank you, Rajni. We have also shared the email ID in the chat box in case you're not available on WhatsApp. You can drop us an email on this communication@omicslogic.com ID, and our team will connect with you there. Okay, I think that will be all from the participants, I guess. And okay, thank you. I'll stop sharing then. So thank you so much once again for all the participants for joining with us for this webinar. And we do hope it proved insightful for you. And of course, in case there are still some lingering questions, you may have joined late or you could not watch the recording completely, or there were some internet interruptions, please feel free to connect with our team on the provided contact numbers or the email ID, and our team will assist with you with further details. Thank you so much once again, and we look forward to seeing you in some of the upcoming programs or events. Thank you, everyone. Have a great day.